Hey everybody, welcome to part 2 of Muscles and Muscle Physiology PowerPoint. This is Christina Howard and this presentation is for Biology 241 and this is part 2 of the muscle presentation, primarily again focusing on skeletal muscles and striated muscles as well, of course, because like skeletal muscles, cardiac muscle is striated, but we're focusing mostly on skeletal muscles this time because, of course, the heart gets its own special chapter. So, circling back, in the last presentation, I explained the molecular specifics of how a muscle fiber actually gets shorter as a result of motor neuron excitation being turned into kinetic motion of sarcomeres getting shorter. So if you haven't watched that yet, don't watch this first. It'll only confuse you. Go watch how sarcomeres work and then come back to this later. So tension production is the result of all of the myofibrils in a single fiber being activated. So as a result of the action potential arriving at the neuromuscular junction, it's not like one group of myofibrils in a muscle fiber can get activated and the rest will stay dormant. If there's an increase in calcium in the sarcoplasm, all of the myofibrils in that particular fiber will contract. So from that perspective, the contraction of an individual fiber is kind of all or nothing, where all of the myofibrils are doing it or none of them are doing it. There's no like in between mode. So tension production is the result of individual fibers contracting, and if enough fibers within a muscle contract, then we can get the contraction of the entire muscle. But there are some steps between these two things that we need to discuss. So, tension, which is the amount of cross bridges. So we can express tension as the amount of pulling power or contraction power. You can express that in a variety of ways physically, like pounds per square inch, etc. But the reality is, when our muscles generate tension, what we are doing is creating and maintaining more and more cross bridges. So more cross bridges equals more tension. So this just recapitulates what I said in the last slide, which was muscle fibers, so cells, they either contract or they don't. So within a fiber, there is no way of partially contracting one group of sarcomeres and not having the other groups of sarcomeres in that fiber contract as well. But remember, it's important to note the scale here. Muscle fiber in this context means muscle cell. So I'm talking about the myofibrils making up an individual muscle cell. So tension in a fiber varies for two reasons. One, sarcomere length when the stimulation occurs. So it's not the case that during relaxation, a sarcomere is going to completely get rid of its zone of overlap. The zone of overlap is going to be at some state of partial contraction. And how much partial contraction there is, how much overlap of the myofilaments there is going to be proportional to the ability of that muscle to generate tension. That's called the length-tension relationship, and we'll explore that. But also the frequency of stimulation. So I mentioned in the last PowerPoint, if you want to maintain tension, you have to keep firing the motor neuron at the muscle fiber and keep releasing acetylcholine onto the neuromuscular junction, which keeps the intracellular calcium concentration high. That's going to determine how many cross bridges we can form. If we stop stimulating, or if the frequency of stimulation drops to 0, we don't get calcium in the intracellular fluid anymore because it gets sucked back up into the sarcoplasmic reticulum, and that eliminates the possibility for cross bridge formation, so tension generation is going to stop. So consider the following three images. In image 1, the fibers are almost not overlapping. So we've got a myosin head and an actin filament, myosin head and an actin filament. It's a very, very relaxed, even stretched out sarcomere. So it's going to be difficult to get this one to start generating tension because the amount of cross bridges you can form at this extremely stretched length is pretty small. At about 50% contracted, you have a lot of cross bridge formation power, which means a lot of pulling power. And at maximum contraction, well, the myosin heads and the thick filament end is going to run into the Z disc. So you can't really get any more contracted than this. So if you've already generated the maximum amount of tension, You can't go over that. So this is not going to be able to generate much tension because it's already done. This one's not going to be able to generate much additional tension because there's very few cross bridges that can form due to the spatial arrangement. So the optimal condition for maximally efficient tension generation is this middle state, where there's a resting state of partial contraction. And we call this muscle tone. So if you plot the tension as a percent of maximum, so if this is maximum, how much of maximum have you achieved? Where 100% is it's as contracted as it's going to get, and 0 is not contracted at all. So if you plot as a percent of maximum, we get this curve where the length and the relationship of the length to how much tension it can generate is plotted in this kind of a hump. So down here at 0, we have 0% of maximum ability to generate more tension. This is because we're getting to 0 because we can't generate any more tension than we already have. So if this is a little bit confusing because you're like, hey, this is already maximally contracted, so why isn't it at 100? That's because we're already doing as much tension as we can in this picture, so we can't generate any additional tension. So what this axis is showing you is how much additional tension can you get to from this starting point. And if you're at already contracted, the options for increased tension are 0. So we have this decreased length side where this is sarcomeres very, very short, up to and including as short as they can get. And over here we have very stretched out sarcomeres up to and including as long as they can get without significantly damaging the muscle. So this is an extreme stretching state. This is something that you would basically observe in a laboratory setting where you're putting lots and lots of tension on an individual cell preparation, and not something that you would experience in real life in your own muscles, hopefully, because that would result in injury. So as you can see from this image, the maximum tension you can get additionally is achieved when the sarcomere is about halfway contracted, where the zone of overlap is large, but the thin filaments don't cross the sarcomere center, so we still have a bit to go. If we go to the other side, now we're having a hard time generating tension because the number of cross bridges that you can form in these states, so pretty stretched or majorly stretched, is either very few or 0, and that means that we can't generate any additional tension either, or it's very, very difficult to. So the normal range for the muscles in our body, the physiologically relevant range, so what we would experience in our normal physiology, is about 1.6 microns to 2.6 microns, so only about a micron's worth of difference. These outer limits, these are a result of single muscle cell preparation in an experimental laboratory setting. So somebody stimulated an individual fiber so much that it was maximally contracted, and it couldn't get any more, or they stretched it out so much using weights or pulling that it couldn't generate any tension additionally. So again, these blue zones are outside of what is considered the physiologically normal range. So you don't have to worry about this or this in you for the most part. You're operating in this zone that is purple, flanked by this middle zone of ideal tension. Okay, so let's talk about twitch, and not the streaming service. In physiology, in muscle physiology, twitch is how fast a muscle generates tension in response to a single stimulus. So one motor units, or not one motor units, one motor neuron's worth of a single impulse. So one motor neuron releasing a single impulse and then stopping. So these are again produced in controlled experimentation. So these graphs that you're looking at are graphs of, different muscles, and you'll notice that stimulus happens. So this is the impulse going from the motor neuron to the muscle. Your eye muscles generate maximum tension really soon after the stimulus, but they also lose it really soon. Your gastrocnemius, which is the muscle on the back of your calf, this one generates tension pretty quickly as well, but it takes a little bit longer to decay. The soleus, which is another calf muscle under the gastrocnemius, takes a lot longer to reach maximum tension after the stimulus, and it also takes a lot longer for that tension to decay. So the twitch duration is pretty long. It ranges from as short as 7 milliseconds, so that's like the eye muscle range, to as many as 100 milliseconds, or 100,000ths of a second. So what we're seeing here is after the action potential from the neuron results in acetylcholine crossing the synaptic cleft and binding to the motor end plate receptors that are acetylcholine receptors and also chemically gated sodium channels, how long does it take for that muscle to generate as much tension as it's going to generate from that particular stimulus? So we have the contraction phase, which is up to the peak, and the relaxation phase, which is when it relaxes. So after the impulse has passed and the sarcoplasmic reticulum is sucking up that calcium again, and we have no additional calcium being released. You'll also notice that there's a latent period, which is this short amount of time between when the stimulus actually arrives and when the actual contraction begins. And the reason that the latent period is important is because what this tells us is that contraction follows excitation. They're not simultaneous. I must first experience a graded potential as a result of the chemically gated ion channels opening to let sodium in, a variable amount of sodium. If that stimulus reaches threshold, it's going to take a little while, a few milliseconds, for the graded potential to initiate an action potential and for the action potential to travel down the T-tubules and begin to open the sarcoplasmic reticulum to begin releasing calcium. And once the calcium release has begun, then we get the contraction phase, but not before. And there is a little bit of time there where it takes some time for the events that I just described to happen. So, if we know that a single stimulus gets you one iteration of tension generation, then multiple stimuli in a row, if they're close enough together in time, should get us up to a potential for maximum contraction possibility. And this is across an entire muscle fiber, so one entire cell. So twitch is just one iteration of this. So one stimulus applied and one generation of tension. So here we have multiple twitches in a row. So Trep is the maximum tension, and we get this increase in peak tension after each successive stimulus. So what you see is after the first stimulation, we get a small hump, and then it gets a little bit bigger, and then it gets a little bit bigger, and then it gets a little bit bigger until eventually, as long as the impulses are spaced consistently, the stimulus impulses, we're going to start to achieve the maximum tension each time because the muscle has been primed. But we're not going to get any more than that, and we're not going to add up these waves unless I move these stimulus impulses closer together. So each successive stimulus is delivered shortly after the completion of the relaxation phase of the preceding twitch. So next stimulus arrives once this fiber has already gotten back down to baseline and relaxed from its initial twitch. If the next stimulus arrives before the relaxation period has concluded, then we get what's called wave summation, which is where each peak is higher than the last because we're adding to the height that we gained as a result of the previous twitch. So we get this sort of stair step up where the waves are summed or added up together. So this wave gets partially relaxed, but it is stimulated to generate more tension before it gets to relax completely. So more cross bridges, and then more cross bridges, and then more cross bridges until we get to this peak. And then if we stop stimuli, it will decay back to zero tension after time. So incomplete tetanus is when the stimuli are arriving from the motor neuron so frequently that we get the additive wave summation, but then after wave summation, we're going to get this maximum tension that's been achieved, and unless the stimuli come closer together, we're just going to stay at this sort of plateau here where we have this high tension generated and then only small fluctuations away from that high tension in between stimuli. So it doesn't go back all the way down to zero between stimuli. And that's because the impulses are arriving without enough time for this to decay all the way back down to a completely relaxed state. So the period of relaxation is too brief to let this get back down to baseline. Complete tetanus happens when the frequency of stimulation is so high that the waveforms are eliminated. There is no relaxation phase, so the cross bridges form rapidly and they reach maximal formation rapidly. And then once the sarcomere is all the way contracted and the muscle fibers are all the way contracted, we just can't get any more contracted than that. There's no more options, so we get complete tetanus. Incomplete tetanus and wave summation is what we would see when we're using a muscle and then sustaining an amount of contraction. So if I lifted up a cinder block and then I held it suspended in the air with my biceps brachii contracted, I would see something that looks like this. This situation you would see if you applied outside electrical stimulation to a muscle. So this is, again, an experimental setting. Or if there is something wrong with you where you couldn't turn your motor neurons off and they were just hammering your muscle fibers really, really badly. So you may notice that this word tetanus reflects the name for a particular disease. This is one of the things that happens during the disease tetanus. You get disinhibition of motor neurons, which causes forceful contractions, which cause the muscle tension generation over time to resemble this. That's pathological, so that's bad, and that will eventually kill you. We don't want tetanus. So let's go over these one more time, because repetition is our friend. So during Trep, we've got an increase in intracellular calcium amount because the stimuli are arriving fast enough that in between stimulus events, we are relaxing back down, but there is still some some calcium that is not making it all the way back into the sarcoplasmic reticulum. And so reinitiating another bout of contraction is pretty easy until we just kind of climb up to this maximum. Similar to complete tetanus, this is also not realistic, but an experimental phenomenon. So if you just apply very infrequent stimuli, this is what you get, but you have to do it in a laboratory setting with a single muscle cell preparation. If you increase the frequency of the stimulus, you get wave summation, where the additional amount of tension generation is added to the muscle fiber before it's completely relaxed. So we get more cross bridges and then more cross bridges and more cross bridges. So summing or additive. This is physiologically relevant. If we have incomplete tetanus, we have climbed all the way up to maximum possible tension with only short periods of relaxation possible. So we're not sitting at maximum tension all the time, but we're sitting pretty close to it so that if we continue that same frequency of stimuli, we're going to have this pattern form where there's a plateau at the top, not a flat plateau, but a squiggly plateau. So if you have lots of individual fibers within a muscle doing this, but slightly out of sync with each other, where one set of fibers is doing it like this, and then maybe the overlapping set of fibers is peaking when the others are lowered, that results in smooth but sustained contractions. So like when you're holding a heavy object and you're doing it smoothly without jittering. So complete tetanus is a rate of stimulation that is so fast that we get a total loss of relaxation. And again, this is not physiologically relevant for the most part. So tension in entire muscles is determined by both how much tension is being developed in individual fibers, so how fast the motor neuron is sending impulses at that fiber, and then of that muscle, how many of the fibers are contracting. So are we contracting 50% of the fibers in that muscle organ? Are we contracting 75%? Are we contracting only 25%? So that's going to decide how much tension is in the entire muscle as well. So it's both time-based and numbers-based. So a motor unit is the key to understanding the duality of tension in muscles. So the idea that we have two parameters ruling how much tension we can generate in a muscle organ. So a motor unit is going to be one motor neuron innervating several muscle fibers. So you can see this one here is innervating 1, 2, 3, 4, 5. So this motor unit consists of one motor neuron and 5 muscle fibers. And then recruitment is if you need more attention from that whole muscle, adding more motor units within that muscle to mean that more fibers are contracting simultaneously. So to review, a motor unit is one motor neuron and however many fibers it innervates. So they vary in size. Sometimes the motor units are small, there's only 4 to 6. Other times the motor units are very large, so one motor neuron may innervate up to 2,000 fibers within a muscle. It really depends on the muscle and how finely we want to be able to control that muscle. So recruitment is the process of using more and more motor units and recruiting them as the tension needs change. So for example, if I have this, which is an entire muscle organ, which is the biceps brachii, and I have motor units 1, 2, and 3, where we have 3 motor neurons, and each of them are innervating 1, 2, 3, 4, 5, 6 muscle fibers, for example. Then if I only activate motor unit 1, I'm using 1/3 of the possible tension in that biceps brachii muscle. If I additionally recruit motor unit number 2, so I add motor unit number 2's power to motor unit number 1, then I get to 2/3 of maximum. And if I additionally recruit motor unit number 3, then I get to 3/3 or as much recruitment of tension generation as I can do for this particular muscle organ. So if I have the maximum impulse frequency on all of my motor units, that's going to set the maximum tension I can generate in that particular muscle organ. So if we look at the wave summation and the tension generated, if we want smooth, sustained contraction, so again, like I lift up a cinder block and then I just hold it with my elbows at a 90-degree angle, and I'm doing that smoothly, so I'm not jittering my muscle, I'm smoothly holding it in place, that's the result of my motor units all being recruited, but taking turns reaching peak tension so that no individual motor unit is exhausted. So if we add up the tension for all of these motor units together, here's the tension in the tendon that attaches to that muscle. And that is what we perceive as and experience as smooth muscular activity, so not herky-jerky. So I mentioned earlier that a partial state of resting contraction is what we call muscle tone. And this is based on the length-tension relationship. So remember, we can achieve maximum additional tension if our sarcomeres are partially contracted, so about halfway. So this is why we have some degree of activity in resting muscles. So even if you're not actively choosing to fire off some motor neuron commands voluntarily, your motor neurons are still pinging your muscles independently of your conscious thought to generate a low amount of contraction that just keeps the tension in that sweet spot for the length-tension relationship. The better your fitness is, the more muscular action you do, the more you lift weights, the more you move your body, the better your muscle tone is. And muscles with higher tone are healthier. So if there's little tone, you have limp, flaccid muscles, and this also places you at risk of atrophy, where your muscle fibers individually get smaller because they lose sarcomeres due to disuse. Moderate tone results in firm, solid muscles that are rich with sarcomeres. There's lots and lots of partial tension, and this gives them a greater ability to generate tension more quickly if asked to. So that's kind of what muscular fitness is about. Now, that doesn't have anything to do with aerobic fitness yet. This is just about tension possibilities. So why is muscle tone good? Well, we learned about joints, specifically in the more complex diarthroses like our shoulder and our hip joint, that we need both tendons and ligaments to stabilize those joints. So the muscles that connect your brachium or your upper arm to your scapula and your trunk, including the deltoid, the biceps brachii, the triceps brachii, these are going to stabilize the position of that joint. And it prevents changes in position that could threaten the joint. It also accelerates recruitment. So if you have a partial state of resting contraction that is favorable and has good tone, you can recruit more motor units more quickly. It also increases energy usage. So if you have better muscle tone, you're going to be burning more calories even when you're at rest than someone with poor muscle tone. So this is a major argument for if you're trying to lose weight, don't only do cardio. Lift weights, increase your muscle tone because you will use more calories at rest if you do that. And that ultimately impacts your appearance. Okay, so I mentioned the possibility of generating tension without a length change and how we would talk about that later, and now we've arrived at that time. So iso means same as. And then we have tonic versus metric. So let's talk about isotonic versus isometric contractions. So isotonic is referring to tension rising and then reaching some maximum in which that we have enough cross bridge generation where length is changing but tension reaches some plateau. So this is an example of isotonic contraction where we have a 2-kilogram weight being lifted off the ground because this muscle is generating tension and it's getting shorter while it's doing so. So when the tension is greater than the load placed on the muscle, in this case 2 kilograms, we get concentric contraction, which is a physical shortening of the muscle. Eccentric contraction is when you are controlled lowering something. That's when tension is less than load, but you're not completely letting go. So your muscle is still generating tension, but it's doing so while lengthening rather than while getting shorter. So concentric exercises are like doing bicep curls, where you are shortening your biceps brachii, making the angle of your elbow joint more acute, and you're doing so against a load because you're making tension in your biceps brachii greater than the weight that you're holding. Eccentric is the opposite of that. So in between curls, when you controlled lower the weight back down, and you should be doing that, you shouldn't just drop it and go flaccid, you should control the extension portion of your bicep curl, then you're doing eccentric contraction, where you're lengthening your muscle, but you're still generating tension while you're doing that. Isometric, metric meaning length, means we're generating tension, but there isn't a change in length. So for example, if I'm attached to a weight that exceeds the muscle's peak tension capability, I can still generate tension, but the tension's not going to be able to overcome the weight. So I'm going to have this maximum possible contraction without a length change. So this is isometric contraction. So no additional change in length. Okay, so let's talk about lactic acid, because lactic acid is a product of what happens when you basically surpass your available oxygen for the demand that your skeletal muscles are placing. And that creates a situation where you form lactic acid, and we have to do something with that lactic acid because it can't stick around our tissues. It's not good for our tissues if that happens. So in your chapter that you're reading, you're going to read about the series of events that happen when the demand for oxygen of a skeletal muscle begins to outpace the oxygen supply, and the muscle is going to shift over into first using things like creatine phosphate to generate ATP, and then it's going to switch over from aerobic respiration from creatine phosphate into glycolysis, which is, remember, the quick and dirty way to make ATP. It only has a net yield of 2 ATP, which is not very efficient, and it generates lactic acid as a waste product. So the lactic acid is being generated if we're basically using our muscles faster than our oxygen supply can keep up with. In that case, lactic acid is going to diffuse into the blood because it's going to go from an area of high concentration around your muscle fibers into those capillaries where there's a lower concentration of it. And then the liver takes that lactic acid and converts it back to pyruvate or pyruvic acid. So you might recognize pyruvate from the steps from glycolysis through the citric acid cycle all the way to oxidative phosphorylation, depending on how much you remember about aerobic respiration from 160. But remember, pyruvate is the 3-carbon molecule that you put into the citric acid cycle, also called the Krebs cycle, and the Krebs cycle turns around and generates both reduced electron carriers and ATP. So we can reuse this pyruvate that we originally had as a waste product to generate more ATP. Or we can also glue some pyruvates back together to remake glucose for glycogen storage. So the making of glucose from stuff that's not glucose is a magic trick that your liver can do, which is called gluconeogenesis. So that's glucose, new, genesis, to make. So gluconeogenesis is when your liver makes new glucose out of stuff that wasn't glucose before. And one of the ways it can do that is by converting lactic acid from your muscle usage to pyruvate and then taking a couple pyruvate and gluing them back together. So the cycling of this is called the Cori cycle, and it's a critical way that we remove, recycle, and reuse lactic acid. So it's not just peed out like other waste products. We recycle things to the greatest extent that we can because that helps us conserve our calories, which is pretty ingenious. Okay, so hormones and muscle metabolism. What makes muscles get bigger? What makes them grow? And how do they grow? So this is going to bring in some concepts that I mentioned from chapter 4 and some of the growth and tissue shrinkage stuff that I mentioned in the end of the tissues lecture. So if you need a review of atrophy and hypertrophy versus hyperplasia and things like that, go watch the end of the tissues lecture for a review. So growth hormone does what it sounds like. If you're growing, if your skeleton is getting longer, that means your muscle fibers have to get longer, which means that you need to add sarcomeres. So this is going to stimulate the synthesis of myosin and actin, as well as the other contractile proteins. Testosterone also does this. So if we have testosterone on board and high circulating testosterone, it's going to favor even more actin and myosin synthesis. And I'll talk about something that people do about this particular thing in just a moment, but my dog is asking to come inside, so I'm going to pause and let him inside. All right, I'm back. So we talked about growth hormone and testosterone. Note for both of these, all we're doing is increasing the synthesis of the proteins inside of a muscle fiber that are contracting. We are not generating additional muscle cells. So the corollary to that is that the way that our muscles grow once we're adults, so once we're no longer developing embryos and fetuses, is not by an increase in cell number, but rather by an increase in cell size. So we are hypertrophying. We are not hyperplasia-ing, which is a made-up word. We're not performing hyperplasia or a growth by increase in cell number. Thyroid hormones increase energy consumption by muscles. So that's going to increase your basal metabolic rate, for example, but not necessarily increase protein synthesis. And epinephrine and norepinephrine is going to stimulate muscle metabolism. So this allows us to perform those sort of quote-unquote superhuman feats of strength during fight or flight responses that we wouldn't ordinarily be able to perform. So if you hear stories about like moms lifting cars off of babies, for example, which seems hyperbolic and very dramatic, but has happened, and it is a medical anomaly that's been observed, what was happening was that this person was experiencing such a rush of adrenaline that their skeletal muscle metabolism was supercharged momentarily, and that provided them with enough extra tension generation to do something that they wouldn't ordinarily be able to do. So I mentioned hormones and muscle metabolism. So anabolic steroids are a type of testosterone analog usually. So we don't usually use actual testosterone. We use analogs of testosterone or things that are supposed to increase testosterone or mimic testosterone. And people are injecting these things into their muscles in an effort to increase contractile protein synthesis. So hypertrophy their muscles the short way without working out as much by hijacking their endocrine system. That is not without consequences though. So if you look at the way that steroid hormones are produced, and I won't show you this chart right now, but you can look it up on Wikipedia, it's pretty cool. What you'll notice is that it's really easy to convert testosterone into either dihydrotestosterone or back into estradiol. And there's enzymes that do both. So if you overload your body with testosterone or something that increases your testosterone or is a testosterone analog, what you see are side effects. So yeah, the muscles do indeed grow, but you also get horrific cystic acne because a lot of that testosterone is converted into dihydrotestosterone, which supercharges sebum and sebaceous gland production, and then bacteria get in there and cause a cystic acne outbreak. This often occurs on the chest, back, neck, and face of people abusing anabolic steroids. We also get changes in mood and aggressive behavior because testosterone basically amplifies the parts of the brain that are responsible for basal emotional responses like rage and aggression. So those involve the brain region, the amygdala. And a lot of that testosterone is converted by an enzyme called aromatase into estradiol. And that actually does things like shrink the external genitalia. So the scrotum and the penis of people that abuse anabolic steroids long term experience a decrease in size because a lot of that testosterone is being converted into estradiol. And now the ratio of testosterone to estradiol is off, and that creates changes in external genitalia. So all of those are reasons to not try to cheat at muscle hypertrophy by taking testosterone. It's just not a good idea. You can't avoid the side effects completely, even if you do some other stuff that I won't mention because I don't want to provide anybody with a study guide on how to abuse anabolic steroids. But suffice it to say, there's no real way to use anabolic steroids without consequences, especially if you're using them long term. So just don't do it. Just do the work instead. Okay, so speaking of people that are maybe abusing anabolic steroids, this dude probably can't comfortably wear a shirt. That doesn't look fun, but some people, think that this is the ideal body. So this is a result of the extreme hypertrophy. So not due to more muscle cells, but the increased size of the individual myofibrils. So more sarcomeres, more proteins that make up sarcomeres. And this is caused, if you're not cheating on it, by near maximal tension efforts, which reinforce the need to add more myofibrils. So basically, use it or lose it. And if you're overusing, your tissues will hypertrophy in response and say like, oh, we're experiencing lots of tension. We should probably add more myofibrils to deal with that increased demand. And that's what happens. Atrophy is the opposite situation from that. So if neural stimulation is going to maintain your skeletal muscle, this means that even a resting state of partial contraction or muscle tone will provide some of that function. The corollary to that is that denervation, which is the removal of nervous supply, is going to create muscles that are small and flaccid and weak and eventually begin to lose protein because they're not being asked by a nerve to generate any tension at all. Eventually, if this is extreme enough, we can see reduction to fibrous tissue where the body just decides like, it's not useful to maintain this muscle because we're not ever going to use it again, so we might as well just turn it into collagen. So here is an example of muscular atrophy in the legs. Times when you see this in the extreme are, for example, in people who experience a spinal injury and become wheelchair bound and lose innervation to their leg muscles. Over time, their legs will atrophy to the extreme just due to lack of use, especially if they're not stimulated. So part of physical therapy in that regard is continued external stimulation of those muscles to try and stave off atrophy. Another less extreme and more familiar version of atrophy would be if you break an arm or a leg and then you're in a cast for a while. When you get your cast off, depending on the severity of the break, what you might find is that one of your arms is bigger than the other because the arm in the cast atrophied due to disuse, or same with the leg. So this is a kind of atrophy that's not extreme. You do recover from it, but it's a little bit funny looking when you take your cast off and you've got like a little weak stick arm and then another regular arm, right? Okay, so these are things that you can read on your own. They're not going to be included in your sort of testing stuff, but they're things that students often ask about. And it's a pretty short read, so physical conditioning is only about a page long. And then there's a brief clinical note in a little inset that talks about delayed onset muscle soreness, so what actually causes it. These are, again, things that students tend to ask questions about, but we're not going to go into them here because we don't have time. So go ahead and read these on your own if you want. Just understand that I'm not going to test you on them. Okay, So I mentioned before, we are going to talk about smooth muscle, but not that much. And I also mentioned that heart muscle gets its own chapter because cardiology is its own discipline. So if you're curious about cardiac muscle, unfortunately, you're going to have to wait until next term. So smooth muscle, as you know from studying its histology and its locations and functions for lab, is all over the place. So I was very impressed with how many of you managed to come up with correct locations for smooth muscle on your practicals. Nobody really had any problems doing that. And part of the reason for that is because it's really just everywhere. Anytime where you need to have muscle that's not controlled voluntarily, but makes the diameter of a hollow space larger or smaller, for example, you're going to see smooth muscle. So this includes intestines, blood vessels, the urinary system, many, many locations. And it tends to be organized into sheets, bundles, and rings that form either tubes or hollow organs, depending on the application. So here's some teased smooth muscle. This is the smooth muscle equivalent of pulled pork. I don't like this preparation because it doesn't reflect in any way the actual arrangement, which is down here. So we have longitudinally sectioned smooth muscle, which looks like little fishies swimming in a stream, or cross-sectional smooth muscle, which looks about like this. So practicing recognizing this is what's going to allow you to confidently recognize it over and over again. So smooth muscle structure-wise, we've got uninucleate cells that have very little sarcoplasmic reticulum. So we've got most of the calcium coming from outside of the cell. We have no T-tubules to speak of, so there's no descent of that sarcolemma into the inside, which means that the cells themselves are going to be smaller because they need to distribute that extracellular calcium more evenly throughout the cell. So that sets a limit on cell size. We also have no T-tubules, no myofibrils, meaning no sarcomeric arrangement and no repeating arrangements of sarcomeres. And we've got this sort of loose and diffuse arrangement of thick and thin filaments that can interact and overlap, but only when something causes calcium to be highly available and allows it to link up. So there's more myosin heads per thick filament in this arrangement, which creates more potential for cross-bridge formation, even though the myosin heads, or excuse me, the myosin filaments and thick filaments are not that long. And the thin filaments are not anchored at a Z-disc, but rather at dense bodies. So there's no striations and there's also no troponin. And here's the deal with this. It seems confusing because you might be wondering, like, well, don't we need troponin to bind to calcium to remove the tropomyosin from the active site of the actin? And the answer is, yeah, in striated muscle you do. But in smooth muscle, calcium is going to complex with a protein called calmodulin, and it's calmodulin that is forming that function that basically frees up the active site of actin. So calmodulin complexed with calcium is doing that job. So between calmodulin, the calcium source, and the lack of sort of a repeating sarcomeric arrangement, as well as the fact that we've got so many more myosin heads, we can just form more cross bridges per unit of mass than skeletal muscle. So that creates a situation where each individual smooth muscle cell, although it's smaller individually, can form more cross bridges per weight of cell than skeletal muscle. So it is pretty strong. It shouldn't be underestimated. The calcium source is mostly from the extracellular fluid because, again, we've got very little sarcoplasmic reticulum, but we also have intermediate filaments which are joining adjacent smooth muscle cells. And that transmits the contractile forces from one cell to another. So this allows the smooth muscle to contract in waves that are self-reinforcing. So how does this occur? What are the steps? Well, first, as a result of excitation of the smooth muscle membrane, which is not called a sarcolemma, by the way, because this is not a striated muscle. We only use that prefix sarco for sarcolemma, for example, for striated muscle. We do still call it a sarcoplasmic reticulum, though, which is a little bit silly, but that distinction is not super important. As long as you understand that it's a smooth endoplasmic reticulum that is modified to store calcium, you're good. So calcium from the extracellular fluid enters the cytoplasm, and it also leaks out of the sarcoplasmic reticulum. That calcium binds with calmodulin, forming that complex. that complex activates what's called myosin light chain kinase. So myosin light chain kinase actually splits the ATP for the myosin, and that's what energizes the myosin head to reach up towards the active site of actin. So we need this additional calmodulin and myosin light chain kinase series of steps to make the myosin heads interact with the actin, and that's what generates cross-bridge formation. So this changes the latency to contract because there's some additional steps involved. And ultimately what that means is that the contraction of smooth muscle containing structures is going to be contingent upon how many orientations of smooth muscle exist. So how many different sort of views of smooth muscle would you get in a single slice? So just cross-sectional, just longitudinal, a mix of both. That's going to determine what shape the contracted smooth muscle will take. And because there's some extra steps in there, and because the contraction of one smooth muscle is going to increase the likelihood of its neighbor contracting, smooth muscle takes longer to generate tension, and it also takes longer to relax. So waves of smooth muscle contraction take a longer time to form and reach maximum, and then dissipate and reach minimum as well. So this is why we experience things like uterine cramps or abdominal cramping from our intestines come in waves where the pain builds up to a peak due to uncomfortable smooth muscle contraction, and then it dissipates, and it takes a while to dissipate as well, as opposed to being sort of an on and off switch. Additionally, because of the lack of sarcomeres and repeating sarcomeres forming myofibrils, the overlap amount of actin and myosin within a single smooth muscle can vary within a cell. So that means that tension is not length dependent, and that creates additional flexibility in how smooth muscle actually contracts. So this means a couple things. One of those things is that since there's no length tension relationship, stretched smooth muscle can actually do something that striated muscle cannot. And that is, if you stretch smooth muscle, it will recoil and contract harder in response to being stretched. And it can do this at lengths of up to 4 times more stretch than striated muscle would. So this is one thing that's going to come back around to us next term when we talk about the digestive system, when we talk about how, for example, stretching of the stomach results in gastric emptying, and stretching of the intestines results in an increase in moving the food along during digestion. So, regarding control of contraction in smooth muscle, as I mentioned previously, it's not voluntary, and it's not a mixture of voluntary and involuntary like skeletal muscle is. You cannot voluntarily control your smooth muscle. I can't sit here and dilate or constrict my arteries or my airway. It's just not an option. So we have 2 options for contraction, 2 modes. One is multi-unit smooth muscle, where every single fiber is innervated. And 2, we have visceral smooth muscle, which is single unit, and this is where there's a few of the individual cells that are connected to motor neurons, but not every single one. And the way that the contraction spreads from cell to cell is via gap junctions, which remember are little holes that allow one cell to share its cytoplasm and therefore share ions with its neighbor. So that phenomenon where visceral smooth muscle, like in the uterus or like in the intestines, builds up more slowly to maximum tension and then relaxes more slowly is partially a result of the need for transmitting initial excitation from a motor neuron across multiple cells that are not innervated directly via gap junctions. So this creates a difference in speed and a difference in how detailed or delicate of a control the smooth muscle affords. So multi-unit smooth muscle, where each individual cell is innervated, produces smooth muscle contractions that are very detailed and allow precise control. So your iris, which determines your pupillary size, that's one example. So you can make micro adjustments to how much light you let in your eye. Certain portions of the male reproductive tract that help to move sperm along. Sperm doesn't swim out, it gets propelled out. Large arteries, so examples are your carotid arteries in your neck, for example, and your erector pili muscle, so the muscles that pull your hair into goosebumps, for example. Single unit smooth muscle, which we also call visceral smooth muscle, is controlled by, oops, hold on, I'm getting a phone call. Okay, sorry about that. Wasn't expecting my phone to make sounds. All right, so back to single unit smooth muscle, also called visceral. Remember, this is where one motor neuron innervates a few of the muscle cells, but the rest of them are not directly innervated and are therefore going to be controlled by impulses spreading from their neighbors. So these react to all kinds of things, and that includes chemicals like neurotransmitters, also hormones. So there's a lot of hormones that you'll learn in the digestive system chapter, for example, that regulate things like filling and emptying of your stomach and intestines. The concentrations of oxygen and carbon dioxide also influence this, as well as other gaseous neurotransmitters like nitric oxide. Stretching also initiates this. So this is why the arrival of food in the intestine stimulates contraction to move that food into the next sort of portion of your intestine, and also, unfortunately, irritation. So this is why, for example, if you eat the wrong kind of food with the wrong kind of bacteria in it, you can end up with diarrhea because your visceral smooth muscle of your intestine is moving food through your intestine too quickly due to it being annoyed by bacterial toxins, and that creates loose stools. So more than you ever wanted to know about diarrhea, but wait until next term. There's even more. So interestingly, similar to some other populations of cells in your body, including your heart, again, more on that next term, there's pace setter cells that exist in the smooth muscle of your intestine. And a pace setter cell is a cell that can actually electrically excite itself. So it doesn't have a resting membrane potential, it's constantly drifting towards threshold and then depolarizing by itself. This is because it has more leak channels proportional to its sodium-potassium pump than other kinds of electrically excitable cells. So you get lots more details on these next term. That's just a brief explanation of them to help this make sense. So you can look forward to more details at a later date. For now, just think about it as being cells that can self-excite, absent neural innervation, and they can do that because they're extra leaky to ions. So these initiate rhythmic cycles of contraction that spread via gap junctions. So in the intestines, for example, we call this gut motility, which is just the average rate that your intestines experience contraction and depolarize kind of on their own. And then your central nervous system is going to either increase the rate of that or decrease it, depending on which side of the nervous system we're talking about. And we'll get more information on that when we get to chapter 16, which is the autonomic nervous system. So visceral smooth muscle is arranged into sheets to form hollow organs, and it's primarily controlled by the autonomic nervous system and to some extent local forces and factors like hormones and stretching. But the major determinant is going to be whatever the autonomic nervous system, either your sympathetic or parasympathetic, tell it to do on top of whatever it's doing by the virtue of pace setter cells. So that's visceral smooth muscle. So again, there's some aging and muscle tissue questions that students tend to ask, and you can read about them in section 11.7. It contains a lot of interesting information, which is summarized here, but again, not a part of the chapter that I'm going to test you on. Learning the neuromuscular junction and contraction for both striated and smooth muscle is more than enough for one specific chapter. So there's portions of this chapter that you definitely should read because they're of interest to how people work in general, but that I'm not going to test you on because we want to limit the amount of information on one test. So that's kind of the rationale here. All right, and that's the end of the slideshow. So thank you for your attention. I'm sorry for the interruptions, and I will edit this and post it